In this video, I want to help you understand how proteins inside the cell are actually made from the instructions in the DNA, the genetic code, the genes, the sequence of nucleotide bases. How is this used to actually make proteins inside the cell? So how do we go from genes to the structure of a protein? Proteins such as enzymes, proteins such as hormones, proteins such as antibodies. That's the question we need to answer. To help us understand this, we first of all need to know what proteins are made of. Proteins are made of little subunits called amino acids. An amino acid is a building block of a protein. The cell uses the genetic information in DNA to put a particular amino acid into a certain place in the chain of amino acids. So the instructions in a gene will be used to place an amino acid next to another one in a certain place. There are 20 different amino acids used inside a cell, and their position in this amino acid chain is very important. And this continues and continues until a protein is made. So to summarize, a protein is a long chain of amino acids. The position of each amino acid is determined by the gene. So let's go back to the question again. How is this information in a gene used to actually place the correct amino acid in the correct position to make a protein in a cell? Let's take a sequence of DNA, a sequence of the nucleotide bases, in other words, a gene. For simplicity, I'm taking a small section of DNA in this example, running from bottom to top, CGA, ACC, CGA. What's important to understand is the information comes in groups of three nucleotide bases. This is known as the triplet code, 3 bases triplet. That triplet, which you see in example now, CGA, is the information a cell uses to place a particular amino acid into the correct position to make a protein. One triplet code results in one amino acid being put in place. Then the cell will use the next sequence of nucleotide bases, the next group of 3, the next triplet code, and so on, until the gene is ended and the chain of amino acids has ended. So let's see how that happens. So the gene will take the first triplet code, so the cell will take the first triplet code of the gene and put an amino acid in place. The next triplet code is different to the previous one, so you'd expect a different amino acid to be put in place, and those 2 amino acids will join up. The next triplet code will be used by the cell to put another different amino acid in place, and this will continue to form a long chain of amino acids which eventually go on to make a protein.